Okay, good morning, everyone.
Good morning.
Morning. Great. So hope everyone's okay. Can you hear me okay? Can you see me okay?
Yes.
Yes.
Okay, great. So let's get started. So today we're going to talk about stem cells. And so I kind of guess you all remember the definitions of potency. So stem cells can have multiple potencies. Can someone tell me what potency relates to? Definition of potency. Wow, really is Monday morning, isn't it? Okay, so potency is okay.
Like how many cells?
Okay, Shiva.
The scope or when a cell divides, what is the range of cells it can produce?
Yeah.
That's
Yeah.
somewhat.
Yeah, that's essentially it. Yeah. So Shiva's right. It's kind of what types of cell types a particular cell can give rise to, and so stem cells can range from many different types of potencies. They can be totipotent, and in this respect, then the zygote is then a stem cell, or they can be pluripotent, which is they'll give rise to a number of structures, multipotent, and so on. Okay, so the properties of stem cells are listed here. Okay, so probably the most important property of a stem cell is, of course, apart from its potency, is that it self-renews. Okay, and so in this respect, the population of a stem cell does not diminish. Okay, and so here you can see this kind of vague diagram here, where a stem cell can divide symmetrically to produce two daughter stem cells. You can also get asymmetric cell division. Okay, and so the stem cell can divide once, one daughter giving rise to another stem cell, and the other daughter then becoming a progenitor cell. And so here the kind of nuance of what a progenitor cell is as opposed to a stem cell is that the progenitor cell has a limited lifespan. So it will divide once, maybe twice, before settling into differentiation. Okay, so as I said, they can give rise to at least one type of tissue as well as the daughter stem cell. And so in this respect, a stem cell is kind of bipotential. That's the lowest potential that a stem cell can have. It can give rise to two types of cells, the differentiated cell type as well as itself. Okay, a progenitor cell we can start thinking as more unipotential. Okay.
Raj?
Yep.
It's Akash. I had a doubt.
Yeah.
So besides the difference in lifespan, what are the other differences between the progenitor cell and the stem cell?
Yeah, so the other difference between a progenitor cell and a stem cell is essentially the progenitor cell can't. I think it's only lifespan, and so it's more of a semantic distinction actually. You know, what's not to say that at this point, this progenitor cell, is not a stem cell because it can give rise to a precursor of a differentiated cell. And so this has always kind of, I mean, if you want to set up, if you want to have an argument with a stem cell biologist, you can ask whether they're talking about a stem cell or a progenitor cell, because this excites a lot of emotions. I think it's generally lifespan, okay? And so you hit the nail on the head when you said, when you talked about lifespan, okay? Progenitor cell is not long-lived, okay? Progenitor cells will become depleted after a short time, or even not necessarily such a short time, but they'll become depleted as they differentiate into their differentiated cell type. Whereas stem cells will not become depleted. Okay.
Okay.
So there is this idea of kind of I mean, maybe some of you know what determines how many times a cell can divide. So anyone want to pull up that fact?
Length.
Sorry, who said that?
Anandita here.
Oh, yeah, go on.
The telomere length.
Yeah, exactly. And so it's possible that stem cells may have ways of resetting their telomeres so that they can divide more. They can divide much more than a progenitor cell. And so, I mean, you can do some investigation of yourselves. Check PubMed or Google and see if that's actually the case, okay? Cool. So kind of one of the classic stem cells is the hematopoietic stem cell. So of course that, sorry, I should finish this. So stem cells are of course associated with a niche, okay? And we'll come back to what a niche is in a couple of slides. So I will talk predominantly about embryonic stem cells, which are the more fascinating cell type, I think. Yeah, but of course, stem cells were initially characterized in adult systems, and the hematopoietic stem cell is a classic example. So adult stem cells are used in normal homeostasis or injury repair. They're usually lineage restricted. And so, as I've said here, the classic example is the hematopoietic stem cell, which is found in the bone marrow. And so hematopoietic stem cells, of course, give rise to all of the cell types within the blood. They show very classic differentiation patterns with a hematopoietic stem cell expressing or not expressing these markers, transiting to another cell type that expresses a different battery of markers, as that then transits to a kind of progenitor cell type, or for example, a lymphoid progenitor, or ultimately erythrocyte progenitor, myeloid progenitors. Okay, so this is kind of the classic stem cell system. This was figured out by Tiller McCulloch in the 60s, but the hematopoietic stem cell, HSC, was originally described over 100 years ago by Pappenheim in Berlin in 1896. And this was purely just by observation. And so he was looking at bone marrow and noticed these cells which are able to give rise to different lymphoid progenitors. He hadn't quite worked out that they were also giving rise to myeloid progenitors as well. But the hematopoietic stem cell was unequivocally identified by these classic experiments from Tiller McCulloch in Canada. And so what they were doing was they knew that a particular dose of X-ray radiation on mice would kill the mice within 30 days because the mice no longer had the ability to fight infections. Their hematopoietic cells were being exhausted. And so they wanted to know what was the minimum number of bone marrow transplant, the minimum amount of bone marrow transplant that would rescue this effect. And so what they did was to irradiate the mice and then inject them with particular concentrations of bone marrow cells. And what they noticed were soon after they would start noticing nodules on the spleen, so these little bumps on the spleen. And when they looked at this at these bumps, they realized that these bumps were full of hematopoietic stem cells. These hematopoietic stem cells were able to be cultured, and each nodule was the result of a single hematopoietic stem cell. And so they went on to show that just these isolated nodules were able to rescue the effect of X-ray radiation on mice. Okay. I don't want to talk about this so much because we've got a lot to get through, but if anyone has any questions about this, please ask now. Otherwise, I'll direct you to this chapter on the history of hematopoietic stem cells, which I've linked down here. Okay. Great. Good. So let's bring in the idea of niche now. So probably many of you know what a niche is. And so does anyone want to explain this for me? Okay. Sure. I'll do the explaining then, okay? So the niche is a microenvironment that governs the behavior of stem cells. And so as I've said, adult stem cells kind of are generally quiescent, which means that they're either not dividing or dividing very slowly. And when they're needed, they'll undergo this asymmetric cell division to give a particular cell type. What governs that behavior, what tells the embryonic stem or the adult stem cell to either not divide or divide very slowly, divide and differentiate, is the microenvironment that these stem cells are in. And so the microenvironment is made up of particular signaling factors, and it could be any signaling factor. There's no one signaling factor that's important here. It's also made up of the extracellular matrix, the substrate that the stem cell is sitting on also affects greatly what the behavior of this stem cell is. It can be also the metabolic activity around the stem cell, and all of these are kind of ways of that regulate stem cell activity. In particular, this idea of renewal versus differentiation. One thing I wanted to bring up, which I think you'll hear in lectures subsequently, either from Tina or in the cell biology course next year, is the idea that immune cells, especially macrophages, can also contribute to niche behavior, in particular subset of adult stem cells. And so what you're looking at here is the characterization of the niche for hematopoietic stem cells. And so this is a bone, this is the marrow. So you'll have these kind of specialized osteoblasts, so bone cells, and these are particularly shaped. They're spindle-shaped N-cadherin positive osteoblast cells, and they generally are in close association with quiescent hematopoietic stem cells. And it's known that all of these particular interactions with all of these particular genes, for example, integrin and VCAM, and N-cadherin interaction, and so on, are important for maintaining the quiescence of hematopoietic stem cells. When any of these are disrupted, the stem cell is activated and undergoes then directed proliferation to form various components of the blood. So that's one niche. And of course, there are many niches. There's one classic niche is the one that regenerates hair. In some of us, unfortunately, this is not working so well, but all of you are young, and so your hair stem cells are still very active. And this is a small population found on the side of the hair cell shaft. They're called bulge stem cells, and they're again maintained by these particular signaling molecules, and in a particular, they're regulated by mechanical influence, by metabolic influence, by hormonal influence, as well as these direct signaling molecules to start enabling differentiation. And so when these stem cells, which are generally quiescent, are activated, they'll migrate down to the base of the hair and start to proliferate to start forming a new hair shaft. Okay. So this is the hair stem cells. We also have stem cells that govern skin, for example, the small intestine. So as you know, the cells of the small intestine are undergoing constant recycling, and so there's stem cells in the base of the crypt of the small intestine that enable this. And as Arjun will talk about later on in these lectures, there are stem cells in the lungs as well, which enable repair of damaged lung epithelium. And these stem cells are also governed by a particular niche that they're in, okay, which depends very much on interactions between stromal cells. Stromal cells mean the mesenchymal cells that underlie this, basement membrane, and so on. Okay, so that's the niche concept. Okay, any questions up until here? Great. So let's go on then to embryonic stem cells. And so, of course, embryonic stem cells are a classic stem cell. They are characterized for being able to give rise to every single part of the embryo. And what I want to do for the rest of this lecture is to talk about ways of thinking about their pluripotency. So this is kind of the history of ESL research. Okay, and so the history is kind of, I mean, when you start looking into history, it's kind of, you realize that everything we do is built on things that have gone on before, and stem cell history is no different. And so the stem cell, the history of embryonic stem cells probably started way back in 1954 with the observation that a particular mouse strain developed testicular teratomas in 1% of males. They realized that these, so what, does anyone know what a teratoma is?
It's a tumor. It's a cancerous tumor that has many different types of tissue in one tumor itself. Yeah,
exactly.
Like hair and teeth and some of those.
Yeah. It's the most monstrous of all tumors, I think. I mean, all tumors are monstrous, yeah, but the teratoma does contain hair and teeth and various ducts and things like that. It's really horrible. Do not look up teratomas on Google today. And it can be, in this mouse strain, 129 mouse strain, it's a testicular teratoma. You can have ovarian teratomas as well. And so the idea that this tumor could give rise to many different cell types started kind of this concept that you could get pluripotent cells generated kind of experimentally. And so these testicular teratomas were taken and cultured. You were able to differentiate them in vitro as well, which was very nice. So experiments could be done on these teratocarcinoma cultured cells. The research carried on, and it was basically found that the embryo, when you took an embryo, a young stage mouse embryo, and implanted it into the testicles of a mouse, that would also form a teratocarcinoma. And so this started giving the idea that the embryo and these carcinoma cells had maybe a similar kind of potential. And these embryos which are forming teratocarcinomas on either extrauterine sites or extra testicular sites were developed into what are called embryonal carcinoma cells, EC cells, and it was found that these EC cells could be cultured in particular media conditions. They could be isolated, and they could be injected into the blastocyst of a mouse embryo, and they will contribute to the mouse. And the contribution is normal. They do not form ectopic teratocarcinomas. They'll contribute normal tissues to the mouse embryo. So as these concepts developed, the idea of isolating actually then the early mouse embryo blastocysts and asking whether these could be cultured indefinitely started to become more attractive. And so this gave us mouse ES cells, and I'll go into how mouse ES cells are derived in the next couple of slides. And so one big problem between embryonal carcinoma cells and mouse ES cells was that the carcinoma cells would never contribute to the germline. And so even though they contributed to a lot of the embryo, they could not contribute to the germline, but mouse ES cells could. And so this allowed many, many different techniques to be able to be set up. If you can culture these cells, manipulate these cells using kind of gene targeting, kind of transfecting in plasmids, using homologous recombination, you could start manipulating the genome of these cells. And if you could make chimeras of these cells and introduce them back into a blastocyst, you could start making knockout or transgenic mice. And this happened in about 1989. First genetically modified mouse was generated by Mario Capecchi, and this kind of gave rise to a whole slew of different techniques using mouse ES cells. We end this kind of timeline here with the derivation of human ES cells, and so human ES cells were derived first in about just over 20 years ago, and these have been used for particular techniques for understanding differentiation of human embryonic tissues as well. And so, and of course, these are now being used to try and repair injuries or replace particular organs using what's now called cell therapy. Okay. So this is the history. Again, I'll direct you to this review, which is from Davor Salter, who was very active during this time in understanding teratocarcinomas and EC cells, and it's a very nice review. So what are ES cells? So embryonic stem cells are essentially all they are. So embryonic stem cells are the inner cell mass of the early blastocyst cultured. Okay, so what you're looking at here is a figure from, I don't have the reference here, sorry. I'll upload the reference later. What you're looking at here is kind of the different ways of making embryonic stem cells. And I'll go into this in a little bit more detail in the next slide. But what you essentially do is isolate blastocysts, either early or late blastocysts, and put them on a Petri dish, and that's it. You put them on a Petri dish, you let the blastocyst stick onto the dish, the cells will start to form colonies, and those cells can then are essentially ES cells. Of course, it's much more involved than this. You need to be able to propagate these embryonic cells so that they can retain their pluripotency. And we'll talk about that in a little bit. Okay, so deriving ES cells is actually remarkably easy. You need an embryo, you need to put it on a dish, and you need media that allows you to retain pluripotency. Okay, it sounds easy. Of course, that last step is what's most troublesome. Okay, so in effect, what you do is, and this is the same for both human and mouse ES cells, is you isolate the inner cell mass of the blastocyst embryo. Can anyone remind me what the inner cell mass is of a mouse embryo or a human embryo?
It is basically that gives rise to the 3 germ layers later.
Yeah, exactly. So it gives rise to the embryo proper, okay? And so as well as the inner cell mass, what else do you have in the embryo at this stage?
The trophoblasts?
Yeah, exactly. Thank you, Kanika. Trophectoderm, trophoblasts, yeah. And these are these cells here, and they'll contribute to the placenta, okay? And so you culture the inner cell mass on this, this layer of mouse fibroblast feeder cells, okay? And this, somehow this emits the nutrients that are necessary for the inner cell mass to grow and to retain pluripotency. Once you have, once the colony has taken hold, it grows a little bit, this colony can be lifted off, dissociated and then replated, and you start getting then propagation of the embryonic stem cells. And so by this point, we're calling this an established embryonic stem cell culture, okay? The media composition, so as you know, there's a kind of media layer on top of this. And so the media composition is actually what's important for this propagation of the embryonic stem cells. And I'm going to talk about the media composition a little bit, and I'll with a particular point. So mouse ES cells, depending on when they're gathered and how they're cultured, can have these 3 different types of pluripotency. They can be naive, ground, or primed state pluripotency. In the classic ES cell derivation technique, what happens is that the mouse is superovulated, fertilization happens, and then you induce diapause by inducing this estrogen analog. Can anyone tell me what diapause is? Okay, so diapause is, go on, Atal.
It is basically some type of arrest which happens during the cell growth.
Yeah, close, thanks. So what it is when we're thinking about mammalian embryonic development, diapause, of course, is is as Atal says, it's actually an embryonic arrest. So the embryo stops developing for a little while for whatever reason. Rodents will do diapause, and so they will arrest the development of embryos if the mother is already suckling. And so, of course, if the mother is already suckling a previous litter, then she doesn't want to have to deal with another litter, and so the kind of burst of estrogen that's in the mother will prevent the embryos from developing, will pause embryonic development. And so once suckling is finished, then the embryo can continue to develop. So diapause happens generally at E2.5 in mouse development, which, as you all remember, is kind of pre-implantation. And so the embryo is not implanted. And then once conditions are favorable, embryonic development can restart. Okay, and it appears that diapause is probably one of the reasons why mouse ES cells are so readily derived. Other mammals do not do diapause. Rodents do, and particular strains of mice do it much better than other strains. And those strains are the ones where ES cells are very, very easy to derive. And so you collect the diapause blastocysts after 4 days. You essentially just plop them down on this plastic plate that has a inactivated embryonic fibroblast layer, and add LIF and a particular serum. So serum is plasma, basically. So it's the spun out components of blood, and this is generally fetal bovine serum, which is used as a way of just getting all the factors and nutrition necessary for cell growth. As well as this particular growth factor, LIF, is needed. And so LIF with serum propagates mouse ES cells, and it propagates them as naive state mouse embryonic stem cells. So if you collect the embryo a little bit later, at 6.5, and then just grow the epiblast, this can also be readily made into a cell culture. Now these are called epiblast stem cells, and they are cultured very easily in this condition. You also have a second condition here, which you're seeing, which is 2I plus LIF, 2I plus SB plus LIF. These are particular factors that are added to the media. And what these will do, they inhibit particular signaling proteins. And so what you have here is the ability to derive embryonic stem cells from any particular mouse, whether it's undergone diapause or not. And this allows you then to induce a type of naive state pluripotency, which is distinct from naive state. It's more induced than naive state, and so they've termed this ground state. Okay, and I'm directing you to this this article here if you want to have more information about these 3 different types of pluripotency. What does all of this mean? So naive state pluripotency corresponds to the early epiblast. So before implantation, it's the inner cell mass. The colony appearance is compact. These particular sets of genes are expressed. Next week, we're going to talk about X chromosome inactivation, but in naive state pluripotent stem cells, both chromosomes are active, both X chromosomes are active when the stem cells have come from a female embryo, which implies that it's at a much earlier state. Okay, you can clone this very easily, so you can take individual cells and they'll grow very quickly. You can target genes in this cell type very easily. So it implies that the chromatin is more open than other cells. And when you inject these stem cells into embryos, it will contribute very highly to different types of tissues, including the germ cells. So the prime state pluripotency is a much more restricted type of pluripotency. It's come from epiblasts, which is very clear. There's a particular type of colony appearance, particular sets of genes that it expresses. Only one X chromosome, so one X chromosome is inactivated. It has low chimera contribution in mouse blastocysts, implying that its potency is much more limited than naive state pluripotent cells. And it has different dependence on different growth factors as well. Okay. So the idea that signaling is involved, so if you remember, you can make something that shouldn't be so pluripotent quite pluripotent by adding these inhibitors. And these inhibitors are inhibitors of particular signaling pathways, FGF and Wnt signaling in this case, or FGF, Wnt, and BMP signaling in this case. And so the idea that signaling is involved in somehow modulating pluripotency starts us thinking that maybe where you have signaling networks, you also have transcription factor networks as well. And so when we look at the type of transcription factors that are define all of these particular steps, we find particular networks of transcription factors that are found either in naive pluripotency. At this point, this is called reversibility phase. This review is from 2014. These two are now ground state pluripotency, and this is primed pluripotency. And you can start seeing that there are these complex networks of transcription factors that are necessary to maintain pluripotency, and by altering the transcription factor network topology, we can start transitioning the cells between all of these different types of pluripotency. And so again, we're coming to a common theme where upstream signals are activating downstream transcription factors, which are organized in some network, and that network ensures robustness, but also some level of switchability. And so by changing some of this network, you can start changing the behavior of the cells, okay? And in this case, the behavior of the cells is what cell types it's able to differentiate to. So one question really is then, what does pluripotency mean, and what does the alterations of these different states actually mean? What could be the mechanism behind it? And so I'm leaving that as an open question. If anyone has an idea what it could be, and what you would want to look at first if you're thinking about, say, a naive state embryonic stem cell and an epiblast stem cell, the primed pluripotent stem cell, what would you want to look at? What would you think is defining its potency? Does anyone want to hazard a guess?
Maybe certain protein factors
Yep.
that are being expressed at specific stages, and then later the expression is stopped.
Okay.
So leading to change in potency.
Okay. So how do you think that would work?
Essentially, maybe initially, or the entire genome is active.
Yeah.
So that time the factors are being expressed without any problem, and then maybe later some kind of methylation is happening on the genome that is specifically restricting the factor expression. I don't know, I'm not sure.
Yeah, no, that's a great idea. Yeah, so this idea of perhaps epigenetic regulation, kind of
Yeah.
restricting the possible outcomes of a cell
Yeah.
is probably correct. Yeah, and yeah, that it'd be kind of fascinating to start understanding all of that.
Yeah. Thank you.
Yeah. So if you remember from the last lecture, we were talking about factors that act to prime condensed chromatin, and factors that will alter will displace nucleosomes, for example. And probably this is kind of what we're thinking about. As well as displacing nucleosomes, we're starting to think about recruiting nucleosomes to limit the number of outcomes from the genome nuclearly that a cell could possibly take. So this gives a kind of very restrictive view. What we're thinking about then is, as development proceeds, we're getting, of course, a gradual restriction in the number of outcomes that a cell could take. Yeah, so we're getting a restriction in potency. What I'm going to do now is to flip this on its head and tell you about 2 experiments that suggest this doesn't need to be the case. Before I do that, I mean, can anyone guess what I'm about to say? What 2 experiments I'm going to talk about? Okay. Can you hear me?
Conversion of adult stem cell into the pluripotent cell.
Yeah.
Yeah, the Yamanaka factor experiment.
Yeah. Okay, that's one. Okay. Cool. So I'll talk about the Yamanaka factor experiment. So what's called induced pluripotency in a little bit, but I want to tell you where those ideas come from. And so, as you all know, and we talked about this very early, that when we're thinking about this idea from Waddington of an epigenetic landscape, we're thinking about the pluripotent cell as development proceeds, heading into a differentiated fate. Yeah, and so we know that in normal development, this will take place. Okay, you go from pluripotency and a gradual restriction in potency until you get differentiation. Every now and then you'll get transdifferentiation. So we know that, for example, if you put particular genes in a fibroblast, you'll get a muscle cell. In a myeloblast, you'll get different cells, so lymphoid cells, and so on. Okay, and so every now and then you're able to cross this valley just by putting in a particular factor. It's also known that this differentiation process, in some cases, can be reversible, and so of course we have many regenerating organisms, planaria, for example, the newt limb, the newt tail, which will regenerate. They will, you can cut up a planaria and it will grow all of the structures in the body. But also you can do these 2 different things, and I'll talk about somatic cell nuclear transfer in the next slide. But you can also fuse a cell to a pluripotent stem cell, and that will reprogram it. That will enable now this differentiated cell to explore different pathways, different differentiation valleys. Okay, let's talk a little bit about somatic cell nuclear transfer, and this is one of the first ways that it became clear that you could that once a cell was differentiated, it wasn't necessarily a one-way trip. You could reverse that information as well. And so these are experiments done by John Gurdon in the 60s. And what he did was to take an intestine from a frog, isolate its epithelial cells, and then suck out one of its nuclei. he put this nuclei in a recipient egg where he removed the nucleus already, and what he found was that a complete embryo could be formed. So this complete embryo, so he knew that it was an embryo from the recipient nucleus, because this intestine had come from an albino frog, and the recipient egg was a normally pigmented frog. And when these embryos developed, the recipient was completely albino. In some cases, when he got just a little bit of the embryo developing, as you do sometimes, he was able to graft this tissue and put it in a host embryo, and find that he would get complete integration of this graft into the embryo as well. And so this gave us the idea that there's the ability of cells to be reprogrammed, to go back to their pluripotent state exists, and somehow the factors that will reprogram a nucleus from a differentiated cell to become undifferentiated, to become pluripotent, exists within the egg, but the DNA itself can be affected to be reprogrammed. Okay, and so this is reprogramming. This was kind of exemplified by the experiments of Yamanaka, who discovered induced pluripotency. And so what he found was that you could put in these four genes, okay, and which are now called Yamanaka factors: OCT4, SOX2, KLF4, and c-MYC, into any somatic cell, fibroblast cells, adipose cells, neuronal cells, and then after some time, at some level of efficiency, these will start forming pluripotent cells. When Yamanaka did this initially, he had to culture he was using mouse fibroblasts. He had to culture them for, I think, 3 or 4 weeks. And he found that his efficiency was between 1 and 0.1%. So only 1 in 1,000 of those cells that he was able to transfect developed as pluripotent cells. Since then, and in the first trial, he wasn't able to get germline contribution. Soon after he did, he was able to make an induced pluripotent stem cell, and by changing the selection media and the way he was doing the experiments a little bit, he was able to then inject these into mouse blastocysts and find that this iPS cell could give rise to every structure in the embryo, including the germ cells. So, of course, in a little bit later, so these experiments were done in maybe 16 years ago, 2004 to 2006. In 2007, Yamanaka and a few others were able to induce human-induced pluripotent stem cells as well, which has set off a huge kind of research field in understanding disease in patient cells, for example, and hopefully repairing particular disease as well. And so this is induced pluripotent cells. I want to finish this lecture just talking about this. So, it's possible to make germ cells from iPS cells. And so the way to differentiate iPS cells into spermatozoa or oocytes is known, has been worked out. And so this kind of gets us into almost science fiction, where we start thinking that, perhaps at some point it may be very easy to take someone's skin or someone's blood, and from there, can we make an offspring of this person just by differentiating sperm and oocytes and recombining them and fertilizing them? So I'm gonna probably not. I mean, apart from the ethical considerations, there's probably other things that are stopping us doing that. But I'll let you think about that. Okay. So that's it. That's all I had today. If there are any questions, so please feel free to ask.
Hi, Raj.
Hi,
I
Raj.
just wanted to know what you mean by feeder cells. In one of the experiments, there was mouse feeder cells in the culture plate.
Yeah. So feeder cells are just a layer of inactivated fibroblasts. Yeah, and so they're alive, but they're mitogenically inactive, and all they're doing is just producing something, producing either the substrate that ES cells can grow on, secreting some factors that enable embryonic stem cells to thrive, essentially. Okay.
Okay. Do they become part of the implanted embryo, or do they just remain as a substrate?
They'll just remain as a substrate. So they're mitogenically inactive.
Okay. Thank you.
Okay.
Hi Raj, I had a question.
Yeah.
The Yamanaka factors are essentially present in the oocyte cytoplasm. Is that right?
So it's not known. It's not clear, and I don't think they are. Yeah. So
Okay.
I mean, equating that. It's a good question. Yeah.
Means are they like being means the oocyte is being produced by the mother's body, so like the mother is essentially giving the factors. Is it like that?
So not quite. Yeah. So it's probably other factors that are in the oocyte. Yeah, and those factors still haven't been really identified. So of course there are there's been a lot of work trying to think about what's actually in the oocyte that reprograms intestinal cells, for example. And there are things like things that strip methylation, things that remodel the chromosomes that are not captured by just OCT4, SOX2, and the other Yamanaka factors. So I think the Yamanaka factors are kind of coming into this in a different way. My feeling is that they're inducing a kind of germ cell fate, and that way you get this kind of reprogramming of the nucleus, which is which then is manifest as pluripotency. Okay.
Okay. Thank you.
Okay.
Hey Raj.
Hi.
Yeah, so I was thinking from whatever you taught today. So basically, we can say that one major difference between a differentiated cell and the stem cell is basically that the network of transcription factors and the genes expressing are they are like the separate groups of transcription factors. Can we say that?
Yeah, absolutely. I mean, you can say that about. You can probably say that about lots, all cell types, and we'll go into this next week, actually. Okay, so next week I want to take all of the stem cell knowledge, think about things like what you're saying, Akash, the kind of transcription factor networks that are unique to particular cell types and changes in that topology, and then think about ways of how to investigate developmental biology. Okay.
Yeah, so I was basically wondering that if we can mimic those expression levels of stem cells into the differentiated cells, so would it be possible that the differentiated cell goes back to the stem cell state?
Yeah, it seems to be. The Yamanaka experiment, the induced induced pluripotent experiments seem to suggest that.
Oh,
And go back. Yeah. Okay.
Raj,
Hi.
few years back, I read Yamanaka's paper. So there are only 4 factors which are responsible for induced pluripotent cells. But the slide you showed, there are several other factors also.
Yeah.
So are those factors also as important as those 4 factors, or they are not that much important, like compared to those 4 factors?
So they so those 4 factors can be, some of them can be interchanged. Yeah. So I think c-Myc, for example, can be changed for other factors. So there's some that are important. It seems that OCT4 and
SOX2.
Yeah, are vital. But the other 2 can be changed for other factors as well. There's a very nice review, and I'll put it up on the reading list a little bit later, which talks about where we can go from here. It talks about chemical reprogramming of somatic cells as well, and those chemicals include things that will strip chromosomes of methylation sites, for example, and things like that. So yeah, I'll put that up. Yeah, and you can have a look and think about these questions. Okay.
Okay. Thank you.
Okay. Okay. No more questions. Cool. Okay. So we'll see you on Wednesday for my last class in this session, and that's it. That's it. Okay. Have a great day, everyone.
Thank you.
Bye.
Thank you. Bye.
Thank you.
Bye.
Bye. Thank you.
Thank you.
thank you.